Hello students. Today we're going to discuss the magic of DNA. When I started in this business in the late 60s, we had various ways of differentiating humans. We had ABO blood testing, which determined whether your blood type was A or B or AB or O. 4 qualifications. The most common of them is type O, and the least common is type AB. But none of them come anywhere close to individualizing. There are other blood types that we've used, such as the RH system, and we began in the subsequent years to do what we called protein typing or enzyme typing. You have a number of enzymes in your body which are slightly different in other people's bodies, like type A and type B. And a number of these we learned how to identify and determine the probability of a random person having that combination of enzymes. That was very complicated and took a long time, and but I did all my enzyme types and my blood type, and I narrowed it down to 1 in 90,000, which we thought at the time was pretty good. But biologists in general thought there are differences in human beings. We ought to be able to identify these differences. So it was a dream that at some point we would have a technique that allowed us to individualize someone from a stain or from his body fluids. In about the mid-1980s, there was a professor of genetics in England who studied DNA, was at the forefront of DNA testing, and discovered that there are certain areas of human DNA that show variation from person to person. In about 1983, there was a murder in that area where he lived. And then about 3 years later, there was another murder. And the authorities developed a suspect, and they did his blood typing, and they did his enzyme typing, and it all matched up. So they were about to prosecute him when they decided that maybe they would go to this professor, whose name was Alec Jeffreys, and see if he could be a little bit more definitive. He agreed to work on it, and he determined that the suspect they had was not the person they wanted, that they had the wrong man, and they subsequently went on a huge blood hunt and got blood from everybody in the area, and eventually Dr. Jeffrey found a match in a man by the last name of Pitchfork, of all names. Pitchfork was convicted and sentenced in 1988, and that was the first use of DNA to identify a person. About that time, the press got a hold of it and coined the term DNA fingerprinting. Well, it's an interesting term. We don't do fingerprinting of DNA. DNA doesn't have a fingerprint. DNA only has sequences of A's and T's and G's and Z's, as we'll find out in a minute or two. But in any event, that's the origin of of the individualization of humans from their DNA. Once the word got out, everyone wanted to try it, and turned out that Dr. Jeffrey's procedure, while brilliant, was quite cumbersome. It required, first of all, that the DNA be separated from the rest of the tissue, and then he had a chemical that cut the strands of DNA, and different people would get different length pieces when it was cut. You can cut DNA with various chemicals. In any event, then he would mark the DNA with radioactive phosphorus, and he would electrophoresis it on a gel plate, and he would get a large number of DNA samples separated. He would then take the plate and subject it to photography methods because the radiation would show up on a photograph as a black stripe. And it turned out that he could identify, or he could differentiate from people to person to person. He formed a company and was selling this procedure, and it was immensely interesting to all of us, but most of us didn't have the opportunity to duplicate it because he owned it, and we didn't have the money to buy it. And it turns out that not every laboratory was able to duplicate Dr. Jeffrey's work. That doesn't mean it wasn't good work, it just means that other labs, for whatever reason, weren't able to do it exactly the way he did. But it was the beginning of the revolution in biological testing, forensic biological testing, not just in humans, but in other animals as well. So newer techniques were investigated. Turns out there are short pieces of DNA in your short segments, not pieces, short segments of DNA in your genetic makeup that have short repeats. Now, DNA, as I'm pretty sure most of you have been made aware somewhere along your education, DNA is a long polymer, polymer being a chain of molecular pieces that form a large molecule. DNA is made up of strands that are connected, double strands that are connected to each other, and there are 4 nucleic acids that make up the DNA. And if you remember gas chromatography, then you can remember what the 4 are, because 2 of them are G and C. So there's guanine and cytosine, and adenine and thymine, GC, AT. T's only A's, and C's only connect to G's. So if you have one of the strands of DNA from a double-stranded DNA piece, you can determine what the other strand has to be by simply, if there's a C on one strand, you know there's going to be a G in that place on the other strand. And the sequence of those A's and T's and G's and C's is what determines your inheritance. It's a little bit more complicated than that. Actually, it's a lot more complicated than that. Certain pieces of your DNA are called genes, and they determine a physiological or physical trait. Other parts of your DNA are operational. That is, they determine when to start and when to stop making that particular strand of DNA, which is a good thing. Otherwise, our fingers would be continually growing and our nose would be continually growing and so forth. And so one trait may be determined by more than one gene, and that's probably more likely than not. In any event, these little short repeats, they may be only 6, 8, 10, 12 pieces long. That is, A's and T's and G's and C's long. But they may be repeated twice, 3 times, 4 times, maybe more. And at this point, we're not really sure what the purpose of them is. We're pretty sure that the ones we look at in our forensic DNA work do not determine traits. And so biologists have simply said that they don't have any use. Maybe that's true. I find it hard to believe. Not much in biology, not much that survives evolution doesn't have some kind of use. We just haven't found it. But in any event, if you have a particular repeat, we call them short tandem repeats, STRs. Originally they were called mini satellites. I think now we're pretty much gone with short tandem repeats. But in any event, when they repeat, that will be a characteristic of you. So you may have a particular repeat that repeats 3 times. Someone else might have one that repeats 4 times, or 6, or 2. And there are a number of these STRs in your genetic makeup. And if you have a number of these STRs that are the same in 2 samples of DNA, then you can estimate how likely that is to occur. And that work was done by lots of laboratories. Certainly the FBI was heavily involved, and this was one time when the FBI was at the forefront of things and did a great job. But STRs rapidly became more advantageous than Alec Jeffrey's technique. They were less complicated. There was no radioactivity necessary. And so instead of 4, 5, 6 weeks waiting for the plate to develop, you could get results in an overnight or a maximum 2 or 3 days. You still have to go through all the whole process of separating the DNA, and that, the DNA, there's not much DNA in your body, really. It's what we're talking about is the DNA in the nucleus of your cells. And almost all of your cells have a nucleus. The main one that doesn't is red blood cells, which are non-nucleated, so they don't have nuclear DNA. But only if you think about it, when you learned about cells years ago, you learned that the cell is a mostly cytoplasm, and only a little bit of it is nucleus, and the DNA is only part of the nucleus. So they take the tissue and they disrupt all the cells except the DNA. That is, they explode them, they lyse them, and then they spin them at the appropriate rate so that the centrifuge pushes the broken up pieces to the bottom of the test tube, and the DNA remains in suspension, or vice versa. So they separate the main part of the cell, and then they separate the nucleus from the DNA. And then once they've got the DNA, then they can begin to analyze it. And originally we used electrophoresis, and we did it on plates, but we subsequently learned that there's a more efficient and elegant way to measure these STRs, and that is called capillary electrophoresis, which is very similar to gas chromatography, except instead of a gas pushing the sample through the column, you have electricity. Now, DNA has it's a molecule that has an electrical charge, and so the positive charges will be attracted to the negative end of the column, and the negative charges will be attracted to the positive end of the column. And they will be divided not by their affinity, but by their size. So the bigger pieces will come out faster than the little. The bigger pieces will come out slower than the little pieces. So little pieces can wind their way through the column much faster and end up being separated. This lends itself to a computer database, and it, of course, is much faster than the old technique. And so at this point, the FBI has identified certain STRs as being discriminating, and they have called them CODIS, C-O-D-I-S, which is the name of the FBI's DNA database system. So if you have 13 CODIS DNA STRs that match, the odds of it being some other person besides the one you're comparing is virtually, non-existent. They calculate them, and they used to give big numbers like 50 trillion or 16 quadrillion or numbers like that. They don't use the numbers so much anymore. They simply say that we have matched X number of STRs, and all these STRs have names that describe where they are located on the chromosomes. And with that combination of STRs and knowing the odds of each one occurring at random, and then multiplying them all together, you come out with enormous numbers that are now generally accepted by scientists and by the courts as having the ability to identify an individual person. And at this point, we have no reason to believe that any two people would have exactly the same patterns, exactly the same repeats. And if we ever do, then that'll tip us on our head, and we'll have to go back to square one. But we do know that identical twins have almost identical DNA and show up in the CODIS system as being indistinguishable. But other than that, we've not found any individuals that would be confused by the CODIS testing. So there we have kind of the current state of affairs, but there's a couple of other things that we need to talk about that are equally important. So we don't have very much DNA in our body, relatively speaking. A little piece in each nucleus of each nucleated cell. So and blood, as I mentioned, doesn't have, red blood cells don't have nuclei, so they don't have DNA. So we need to get a pretty good sample. We need to get a substantial amount of blood before we could do DNA testing. And another one of our dreams, in addition to wanting to be able to individualize someone from their body fluids, another one of our dreams was, lots of times in the crime lab we get a little sample and we say, gee, we don't have enough sample, I wish we could make more. And we used to laugh about that because, make more, yeah, right. Well, it turns out that there is a way to duplicate a DNA strand. The scientists thought about this and they said, wait a minute, your body makes DNA every day. Every minute of every day, you're somewhere in your body, you are reproducing cells. So there must be some way that we can do this in a test tube. And they knew that there were enzymes that catalyzed the reaction for making a DNA strand. We knew that living organisms could do that, and so they isolated that enzyme, those enzymes, and they took a bit of the A's and T's and G's and C's and put them in a pot, and then they heated up the pot. When you heat up DNA, the strands separate, and then if you're going to reproduce them, you need to take the A's and T's and G's and C's and line them up with every G from the separated strand. You'll have a C on the other one, and with every T you'll have an A on the other one. So there's an enzyme that catalyzes that reaction. The problem is that enzyme, the one in humans, certainly, is very subject to breaking down under high temperatures. So they thought, what are we going to do? We can't control those temperatures adequately. And if we put the A's and T's and G's and C's in a pot with some enzyme, and we heat it up and the strands separate, well, that's fine, but the enzyme is now denatured because it was too hot. So somebody with greater insight than me by far thought, wait a minute, there are reproductions going on. There's DNA replication occurring in nature in places that's quite warm. And one of those is the hot pots in Yellowstone National Park. You've probably been there and you've seen the color of the hot pots, and that color is the result of algae that grow in there. So they went to, they got some of that algae and isolated their polymerase. Now, that's polymerase means an enzyme that polymerizes. Anytime you see ASE on the end of a big long word in biology, or particularly molecular biology and biochemistry, then you know that's an enzyme, ASE. So this is an enzyme that catalyzes the polymerization or the manufacture of DNA. So they perfected a technique called polymerase chain reaction, in which they are able to duplicate a strand of DNA by warming it up in a container with some A's and T's and G's and C's, and putting in a little bit of this polymerase, which they get from the algae originally. There's other polymerases now, but the original, and maybe still one of the best, was one that came from the Thermus aquaticus out of Yellowstone. anyway, so you heat that up, and then when you cool it back down, the polymerase facilitates the production of a new strand. So you start with one double strand, and you warm it up, and you cool it down, in which in the cooling down process, it forms a new partner. So now each side, instead of being one single strand, is now a double strand. So you've doubled the amount of DNA with this process. And you can keep doing that, and there are instruments, there are machines that will, you can set them to, okay, we want you to do that 15 times, want you to heat and cool 15 times, want you to heat and cool 20 times, whatever. So it's automated. And so you start off with a very small sample of DNA. And if you double it, then very quickly you reach large numbers. There's the old story about the king who wanted to reward one of his subjects, and he said, what do you want for a reward? I'll give you anything you want. And the shrewd subject said, well, I just want you to get out a checkerboard and put 2 grains of rice in the first square, and in the second square put 4, and in the next 8, and the next 16, and so forth. And the king said, oh great, just bring a sack of grain and we'll do that. What he didn't realize was, by the time he got to the end of the squares, there wasn't enough grain in the world to because when you start doubling, the numbers get bigger very, very quickly. In any event, so now we have a process in which we can make our own evidence. And if done right, it will make exact copies of the original. Now that's one of the great amazements in my life, when I learned about DNA and that it was able to replicate. And you think about how many million times it has to do that. And if it makes a mistake and doesn't duplicate it just right, then the likelihood is that that mistake is not going to be in your favor. And it's called a mutation, and most mutations are not advantageous. So if we didn't have this almost foolproof way of replicating DNA, we wouldn't be able to reproduce, because the first, you'd have a person, but then the next one with a whole bunch of mistakes in the DNA wouldn't be a person anymore. We don't know what it would be, some kind of a blob, but it wouldn't be a human being, wouldn't live. So, this to me is just absolute beyond comprehension most of the time when I think about it. But as far as the forensic scientists were concerned, now we're capable not only of individualizing DNA, but of individualizing very small samples, because we take a very small sample and make it into a big sample, one that we can handle, one that we don't have to worry about just a few cells. Now we have millions of strands of DNA. And so PCR, and of course PCR is not just used in forensics, it's used in medicine, it's used in all kinds of biological science experiments, even the dreaded COVID-19. If you listen carefully, occasionally you hear the doctors talking about a technique that uses PCR. So it's a universally accepted technique, and just enables us to do what we call touch DNA, meaning that when you touch something, you leave a tiny amount of DNA on that surface, and then through the magic of PCR and capillary electrophoresis, we can make enough of a sample. Theoretically, we can start with one cell, but we don't normally start with one cell. Mostly they like to have 10 or 15 or 20 or more, but a very, very, very small number of strands of DNA can be replicated into as many as you need. And that's the secret and the magic of DNA. So with that in mind, we need to talk a little bit about what that means as a practical matter, and we won't go too much more into the actual mechanics of doing the analysis. You can certainly learn those techniques in a molecular biology class or microbiology class, depending on what institution of higher learning you go to. But the techniques are pretty much the same from place to place, except for, of course, you need to be more careful when you're dealing with evidence that you don't get contamination and those sorts of things. But we've learned, because we know the potential for contamination with DNA. Since we're replicating the DNA, the polymerase chain reaction doesn't just duplicate human DNA, duplicates whatever DNA is in there. So if you contaminate a sample, you may you may make new DNA samples that are not made by the suspect. Maybe they're made by you. Maybe you contaminated it. Maybe you sneezed. Maybe you dropped a little blood in it, or whoever. But in any event, we know that that's a possibility, and we have developed techniques to recognize when a sample has been contaminated and know the difference between the main DNA source and the secondary DNA source, which could be contamination. But we're not going to go into much more detail than that as far as the technique is concerned. We're going to talk about the practical aspects of it and what we can do with it and what we can't do with it. So with that basic understanding of how DNA typing works, let's talk about a few of the limitations and a few of the new developments that are coming on in the near future. First of all, remember that the that the STRs, which are the basis of the CODIS, FBI's massive database of DNA, the STRs do not code or do not determine any phenotypic characters. We can't tell the color of your eyes, we can't tell the color of your hair, can't tell how tall you are using the CODIS information. What we can do is we can match samples from the same individual, and the main limitation, of course, is that that individual has to be in the database. If he or she has not committed a crime and has not been required to submit their DNA, they won't be in the database and the computer won't find them. And so that's the thing that the FBI has been working on, but they've been very careful not to accept information from untrustworthy or perhaps unscientific sources. So in order to submit data to the CODIS, you have to be accepted by the FBI as a certified lab. Utah, of course, is a certified lab, and they can submit DNA, but we don't take DNA from everybody we arrest. We only take DNA from certain serious criminals after they've been convicted. Other states take DNA samples under different rules and regulations. So the fears that were initiated by the civil libertarians that we would use the DNA to discriminate against someone or to ultimately use that against them in some way to deny them insurance or something like that. That hasn't materialized from CODIS, and it won't because of the nature of the information that's recorded. Next thing that I think is very, very important is that we remember when we find a sample and have it tested for DNA and the sample matches, that it does not convict anyone. It does not prove that they committed a crime. What it proves is that their DNA was there. We need to be really careful that we know the situation, the circumstances, and is there a legitimate way that that DNA got there. For example, if you go into the bank and you touch the counter, and then subsequently a bank robber comes in and robs the same teller and touches the same counter, they find your DNA on there, and maybe they find his, maybe they don't. But the point is, your DNA is on the bank counter, and that doesn't mean you were the bank robber. And sometimes juries get a little bit confused about those particulars, and they say, well, we've got DNA, we don't need to do any more, we don't need to listen to the rest of the story. I can also say that I've been involved in a couple of cases where, not to say that the person didn't commit the crime, but the DNA evidence was very questionable. We had a case in which a girl was found in a ditch in the western part of Salt Lake County, and they found DNA on her clothing several years after the crime, and they had initially suspected a young man who had witnesses had put her with the suspect earlier in the evening, and he was the last person that they could show had been with her before she disappeared and her body subsequently discovered. Initially, he said that they had not done any serious sexual activity that night, but then when they discovered his DNA on her blouse, he said, well, yeah, we really did. We were making out and we had oral sex, and the DNA was refuted as far as proving that he committed the murder, and he was acquitted. So DNA can be, one of those things that we have to be very, very careful with. I did a little study on another case in which they found DNA on money. The suspect's DNA was on some money that was taken from, or not stolen, but was given as change to some customers, and subsequently they found that the victim's body was in the back room, and they found his DNA on money. Well, I took some money to the Sorensen laboratory that was just randomly selected, and they were able to find usable DNA on every bill that I submitted to them. So when you're handling money, you're leaving your DNA on the money. So once again, we have to make sure that we don't just get the DNA and say, okay, that proves him guilty. I think they did that a lot early on with fingerprints when they said, well, we've got fingerprints, that proves guilt. No, physical evidence doesn't prove guilt. We have to always remember that physical evidence, no matter how powerful, only increases our ability to understand what happens. And we need to get the big picture before assessing guilt or not guilt. And by the way, I don't ever say guilt or innocence, because innocence is not a legal term. It's either guilty or not guilty. It's not guilty or innocent. Lots of people are not innocent, but they're guilty of whatever. So the gold standard right now is the CODIS DNA. It's the largest database. It's the one that most frequently is queried and finds the most hits. But there are other things that are coming on board. There are now techniques, not necessarily discovered or invented by forensic scientists, but there are techniques now that allow us to determine what physical traits are determined by what genes or what DNA sequences. And that allows us to make some generalizations about what the leaver of the, or the person who left the DNA was like. So in cold cases, when we aren't able to identify a suspect, they have sometimes used those characteristics to give a description of the perpetrator. And this is called phenotyping. It certainly is valid and it works, but it's actually quite limited in basically general characteristics. You couldn't, from the phenotyping DNA, draw an accurate picture of me, for example, from my DNA. Someday we may be able to do that, but at this point we can get some basic descriptions and basic ideas, but as far as pinning down a particular suspect, we've got a long ways to go with phenotyping. Another thing that's been making the news lately is familial DNA. If we don't have a suspect, but we have DNA evidence, then one approach has been to see if we can find relatives of that person. So your relatives have DNA which is more similar to yours than to the general population. So in theory, if we could get the DNA of all your relatives, we could say, all right, so the DNA that we found at the scene of the crime is very similar to this group of relatives, and then we may be able to narrow down the possibilities to you. So that has happened a few times. The last time I looked at that, it worked about about 6 times out of 100. So it was. It's not something that I think is going to solve every crime in the world. But so what they do is they go to commercial databases, the kind that you send in your little bit of saliva and $50, and they write back and tell you what your ancestry is. But they have on file your DNA profile. So you can get that from them. They share it, I think commercially, but law enforcement has access to that information. And so they can then query those databases for DNA that's similar to the DNA from the scene of the crime. And oftentimes they will get a list of possibilities. So this person could be related, this person could be related. And oftentimes it's a pretty long number, 70 or 80 or 90 people. And then it's on the shoulders of law enforcement to go and research each one of those and see if they could possibly be the suspect or could possibly be related to the suspect. And once in a while, they get it narrowed down to one or two, and then they hold surveillance on those one or two and wait for them to take out the garbage or to discard a paper bag when they come out of a restaurant, or throw a empty can in the garbage, and then they will retrieve those and get the DNA off them. And if that DNA matches the DNA from the scene of the crime, then they've got a prime suspect, and they'll swoop down and arrest him. That's happened a few times, but think about the probability of it happening, and the probability of law enforcement taking that much time and being that diligent, and that's and working that hard on a particular crime. It has to be pretty serious crime, and circumstances have to be just right. And of course, the suspect's relatives have to be in those databases. So if you're a criminal, it behooves you to tell your relatives, hey, don't be messing around with that DNA stuff. Because they could track you down with that kind of information. It's very spectacular when it works, and when it doesn't work, you just don't hear about it. So that's another area of DNA that's emerging, and should be, I think, utilized as long as we're careful and we don't interfere with people's privacy rights and people's right to not have their DNA inspected by anybody other than the company. At this point, it's still pretty controversial, and the cases where they have been successful have been ruled to be legitimate. But we'll see what might happen in the future. We certainly aren't in a position to predict what the courts will do. Okay, so another thing that has been a glimmer in the law enforcement eye is the time between processing evidence and getting a result. So when we first started out, when we were back in the old days using the Alec Jeffreys methods, it would take weeks to get DNA results, and you needed a pretty large sample. Well, we've eliminated the large sample problem, so the next thing is let's see how short a time period we can get results. So this has led to a whole new field, which is called rapid DNA. And the goal in rapid DNA is to automate the process such that we can get results in 2 or 3 hours rather than 5 or 6 weeks, or it's not 5 or 6 weeks anymore, but at least it's days now. So that's been the goal. And we have some companies that are making equipment that will in fact bring results up in less than a day, just in an hour, a couple hours. And so that brings up the possibility that if it's that fast, and if it's accurate, and if it's simple, why don't we put one in a police car? And then when they arrest somebody, they can do his DNA comparison right there at the scene and get the results and make an arrest. We do that with fingerprints. We submit a fingerprint electronically nowadays to the crime lab, and they oftentimes are able to get a hit through APHIS in a very short period of time. So rapid DNA has a few other problems. First of all, how many police officers have been trained in molecular biology and understand the nuances of really what a match is? That's I don't know that there are very many police officers who are scientists. And so we're putting a very, very technical, highly scientific test in the hands of police officers who, that's their job, to arrest people. And so they have a bias to start with. And then if they're not well trained and they can jump to conclusions, then that can result in arresting people who weren't the guilty parties. So that's one issue. Another issue is the safety of the information. So ideally, if they are at the scene of the crime and they do rapid DNA and they get a result, they would like to get it into the CODIS database. We've already talked enough about that. We know why that would be valuable. The CODIS database is the one that all law enforcement agencies have access to. The FBI is understandably hesitant to accept rapid DNA results into their CODIS. And so they've set high standards for the submission of evidence from rapid DNA. At this point, they have not certified a single laboratory technique or commercial instrument that allows them to submit DNA from rapid DNA analysis. There was one company that was certified and approved, but subsequently they've withdrawn that approval. So at the current time, there's no rapid DNA being submitted to CODIS. That may change. The companies that build the equipment are, they're out there to sell their goods, and they're trying very hard to make it acceptable. So I think that the day will come that we'll be able to do rapid DNA. And the trick with rapid DNA is, instead of having to do all of the steps individually, they sell a compact container that will do the PCR, polymerase chain reaction, and do the separation, all in a little cartridge about that long and maybe that wide. It's absolutely amazing that they can do that, but they have miniaturized it, and so now they're on the verge, I think, of a breakthrough, and we'll probably see it in the next year or two when rapid DNA is part of the world. I'm a little bit nervous myself about having it in police cars. I'd rather see that they take it into the lab and let the lab do it on the rapid DNA machine, and then I feel a lot more confident about the results being interpreted properly. Well, so far we've only talked about standard DNA, nuclear DNA. There are other DNA samples that can be tested. First, let's talk about what we call Y STRs. The Y chromosome is the chromosome that determines male versus female. In the chromosomal makeup of humans, women have two X chromosomes. Men have an X chromosome and a Y chromosome. So we have developed techniques for just testing the Y chromosome. We have probes that only polymerize and mark the Y chromosome. So we can determine, without having the interference from the female victim, her DNA is liable to be the strongest DNA in any sample in a rape case. simply because it's her whole body as opposed to a very minimal amount of fluid or biological material from the assailant. So therefore, that Y chromosome information is particularly handy when dealing with a very small amount of DNA in the victim, or when there are more than one depositor of the stain. If she had consensual sex prior to the rape, then whoever her partner was would have left DNA. And if there are multiple assailants, she was raped by more than one person, then you can have this very confusing mixture of her overwhelming DNA combined with all the DNA samples from the male assailants. So Y probe DNA is relatively specialized. Not all laboratories do it. Requires, while the basic techniques are the same, it requires different probes, and they have to be certified as the laboratory that does Y probe DNA. I failed to mention, and so I'll bring it up now, that the regular STR analysis will tell you whether the sample is male or female. Of course, if it's semen, then we know the answer to that. But turns out that there is a marker, a gene that both males and females have, but they're not the same length. So that particular marker will allow the analyst to say this sample came from a male, this sample came from a female, etc. So they have an unknown sample, and they can at least say, well, this is from a male or female. So YSTRs, back to YSTRs, they're not done in our state lab. They are done in the private labs around, and they are beneficial in lots of cases. I might mention that there's been recent controversy about the methodology for interpreting results when there are multiple donors, and right now it's a bit up in the air. The mathematical manipulations that the FBI used have been called into question, and they have lost some cases as a result of that. So I'm not sure where that's going to go, but right now, if there are multiple donors, we're still in a bit of a state of confusion. Now, there's also another source of DNA, which is not found in the nucleus. Outside of the nucleus in the cells, there are small, what are called organelles, which are simply named for a small organ, and these little organelles, which are responsible for the manufacture of energy in the cell, are called mitochondria. Mitochondria, it's believed, originated as separate organisms in the form of bacteria that have been over the years and years and years incorporated, and now are simply a part of the cell. But they have their own DNA, and is considerably less complicated than the DNA in the nucleus. The DNA in the nucleus has millions and millions of nucleotides in the chain, whereas the mitochondria have way fewer, so much fewer that we're easily able to determine the entire genome of the mitochondria, and we know which areas of it tend to mutate and which ones don't, and of course the ones that mutate are the ones that result in differences. So mitochondria are well studied, and we have pretty good statistics to indicate what sequences are rare and what sequences are not so rare. The big thing about mitochondria that makes them a value in forensics is that they are in much higher quantity. So instead of one nucleus with one set of chromosomes, as in nuclear DNA, there may be dozens, maybe even hundreds of copies of the mitochondrial DNA. And so in samples where there is not much DNA, or it's partially degraded, we can oftentimes still get mitochondrial DNA information. And certainly in things like hair, which are difficult to get the DNA from, the nuclear DNA from, then mitochondrial DNA is oftentimes the only resort. Now, mitochondrial DNA also have an interesting inheritance pattern. It seems that in the development of the embryo, when the sperm meets the egg, in the next development stage, any mitochondrial DNA from the male are destroyed, so that when the child is born, the only mitochondrial DNA it has comes from the mother. And exactly why that is and what the evolutionary value of that is still a little bit in question, but the fact is mitochondrial DNA comes from your mother and not from your father. So if we are able to get mitochondrial DNA, then we can't say this only came from one person, because your mitochondrial DNA is the same as all of your siblings who have the same mothers. They got their mitochondrial DNA from the same mother as you, and since there hasn't been any recombination of the of the splitting in the reproduction process, there's only one female. So all the female mitochondria are the same all through the mother's lineage. That means that if your mother has sisters, then their children will have the same mitochondrial DNA as you and all your siblings. So that greatly reduces the individuality of it. It's still pretty good, and it depends on how rare your particular STRs happen to be, but somewhere in 1 in 100,000 to 1 in 500,000, those are pretty good numbers, but they're certainly not 1 in several quadrillion like we have from nuclear DNA. So mitochondrial DNA, it's DNA, and the same general techniques are used, but we don't have a CODIS database for mitochondrial DNA, so we don't have any actually big databases at all. We can't submit mitochondrial DNA for identification purposes, we can, once the results are known, we can of course contact the FBI and other laboratories and see what their probabilities are, but we can't identify someone absolutely positively from their mitochondrial DNA. And if the suspect has a lot of maternal cousins, and some of those are also, have criminal records or can have somehow been implicated, then that complicates the prosecution considerably, but still narrows it down, and it's very, very valuable evidence and needs to be considered for sure. So let's spend some time talking about collection and preservation of biological specimens. First thing to think about, of course, is the possibility of contamination. And right now we're in the midst of this pandemic, which is why you're seeing me on screen rather than in person. And so we have to make sure we take proper precautions to protect ourselves. And that, of course, means gloves, and if there's any possibility of things being splashed around, or you come in contact with tissues or body fluids or materials that have been handled, that we protect ourselves. So we wear gloves and we wear masks and we wear lab coats, or the equivalent of hospital PPEs, personal protection equipment, when we're for our protection as well as for protection of the evidence. Then, of course, we need to make sure that the collection of it doesn't lend itself to contamination. We need to make sure we don't have a dirty tool that we're picking things up with. If you need to use tweezers or forceps, you make sure they've either been completely sterilized in between uses, or that they're disposable and you've discarded the old ones or saved the old ones with the old evidence, preferably the latter. The next thing that we need to think about is decomposition. All biological evidence is subject to decomposition, and most chemical reactions, decomposition included, take place at under warm and wet conditions. So the important thing is that we don't package wet evidence. And so if we find a bloody towel or a bloody shirt, we need to hang it to dry. Now, if that's going to interfere with the subsequent examination, and you absolutely need to get out of there, then you can package it while it's still wet, but package it in several layers of paper, and then immediately when you get back to your laboratory, remove the paper and hang the article up. Most crime laboratories now have a drying room where they can hang articles of clothing to dry. And remember that drying requires air circulation. So if you package something wet in a plastic bag, there's no air circulation. So we never package biological specimens in plastic bags. We package them in paper bags or boxes, those that, materials that can breathe and let air circulate around in them, or have a fair amount of air in the container. And that's absolute basic minimum, is to make sure that it's able to dry out. Now, for collection of smaller stains, the standard is to use a cotton swab that's moistened with water, with distilled water, and swab it up. And if there's a fair amount of blood, then that works just fine. But it's true also that it's not always easy to get all the sample out of that cotton swab. So if you just have a little smear that you need to take, then you can take one cotton thread and moisten it, and then swab with that in the tweezer and swab up the little stain, and then air dry the thread, and then package that in maybe in a bindle or in a container that will allow it to air dry. There are special swab drying boxes that are available, and if your department doesn't have them, then you should talk to your boss and say, hey, we need some of these drying boxes. And he may not even know what they are, but they're available, and you can take a swab and put them in the box, and they'll still dry. For the taking of standards, if you want, we have to, of course, have a standard to compare to, and the standard way of collecting that is to get what are called buccal, meaning inside of the mouth samples. So you take a swab and insert it into their mouth and rub it against the cheek, and that will rub off enough buccal cells, buccal epithelial cells, to do DNA analysis, and that's a standard collection technique. Again, air dry that swab before we package it as well. So DNA is wonderful evidence, and we all love it, and we all strive to make sure that we collect it properly and carefully, and that we collect the proper standards. Once again, if you're taking a sample from a surface, then take a sample next to it. If you think there's a blood spot there, take a swab of the blood, and then get a new swab, new gloves, and take a swab from next to where the stain was. And that way we know we're testing the stain and not testing the material that it's on. And I think that's terribly important. We had a case a few years ago where a man was accused of murdering his ex-wife. And the main piece of evidence against him was a speck of blood on his sock, on his stocking. And I mean, it was a speck. It was very, very small. And they tested it, and the DNA matched the ex-wife. The problem was the man had lived there off and on for many years, and testified that he occasionally wore his wife's clothing. So if that were a pair of her socks, or if that was a pair of his socks that she had worn, there certainly would have been her DNA on the sock from sweat or cells that slough off or whatever. And they did no controls. And I thought that was a serious defect in the prosecution's case. Because that little speck of blood, while it was big enough to do DNA on, we don't know that the DNA was from that blood. It wasn't like it was an enormous sample and big peaks indicating lots of DNA present, because it was a very small stain of blood. And our techniques are so sophisticated that we can get a result from sweat or saliva or a few skin cells, and we need to make sure that we know what we're testing and that we have proper controls and standards. And I think that I've probably mentioned that a time or two before in this class, but I can't overstate it. And we definitely want to convict those that committed the crime. We definitely don't want to convict those that didn't commit the crime.